this lecture, we're focusing on the magical world of fungi. We're going to talk about their characteristics, their modes of reproduction, their role in pathogenesis and making us sick, as well as the classifications of the fungi. First, we need to cover some definitions and characteristics. The study of fungi is called mycology, and a mycologist, therefore, is a scientist who studies fungi. Fungi is the plural of fungus, and most notably, fungi are not plants. They're a completely distinct group. They're more closely related actually to animals than they are to plants. Our fungi are non-photosynthetic, meaning they do not have pigments to take in sunlight and convert that into energy and food, but they are eukaryotic. They do have those membrane-bound organelles, and they are larger than our bacteria and our archaea. Most fungi are multicellular. So most of us, when we think about fungi, we think about some cute little mushrooms that are grown in our backyard, something like this mica cap mushroom. But notably, there are some single-celled eukaryotic fungi called the yeasts. These yeasts are used often in industry, as we'll talk about in the coming slides, but recognize that we have fungi that are both multicellular and single-celled, individual-celled organisms. So unlike our plants, our fungi do not have true roots, stems, or leaves. There are some characteristics and structures in their anatomy that might superficially look like these structures, but they are not true roots, stems, or leaves. This term is very important. Our fungi are absorptive, saprotrophic chemoheterotrophs. Whew, what does that mean? That is a very, very long description just to say that our fungi feed on dead matter. Okay, the way that they do this is pretty special, and we'll talk about more detail in coming slides, but they actually digest the food first in the environment by excreting these extracellular enzymes that break down that dead organic matter, and then they absorb it into themselves. Okay, that is why they're called absorptive saprotrophic chemoheterotrophs. They're feeding on organic matter that already exists in the environment. They're not making them themselves. Therefore, they are chemoheterotrophs. They have to break it down and absorb it through their hyphae, and they feed on dead things. Okay, so all of that can be summed up by the term absorptive saprotrophic chemoheterotroph. They are able to store their food as energy in their bodies in the form of glycogen. So let's talk about their bodies. Let's talk about their structures a little bit. The main body, the stalk, that is called the thallus of the organism. Then we also have these microscopic tubes or filaments. Sometimes you can see them with your naked eye if there are lots and lots of them. But this is what we call the hyphae. And the hyphae play very crucial roles in the growth and development of our fungi. And we'll talk about that in coming slides as well. Next, we focus on their cell walls. And this is from previous lectures. We learned that fungi have cell walls composed of chitin. C-H-I-T-I-N is pronounced chitin. This is a complex polysaccharide that we also have making up the shells of our bugs and our insects. Okay, so this is what gives the tough outer structure to some of our insects as well as our fungi, believe it or not. Talking about reproduction, the fungi are able to produce both sexually and asexually. They have both sexual and asexual spores, and we classify them by their sexual reproductive structures. And we'll get to that at the very end of our lecture. But just know that their sexual reproductive structures are very important in determining their classification. As you probably know, the fungi are grown best in warm, moist environments, and our spores can come in various shapes as seen here. So why are we talking about fungi? Why are they important? You may not know this, but the antibiotic penicillin is made from a mold, which is a fungus, and that mold is called penicillium. Okay, so here we have something called a streak plate. We have a lawn of bacteria. So this is a petri dish, and what you do is you spread the microbes, the bacteria, all across the petri dish so that they grow up into a lawn. And then when you introduce the penicillium mold to that lawn, it starts producing penicillin. That penicillin is such a strong antibiotic agent that it begins to kill the bacteria that surround it. Okay, so in this petri dish, that penicillium mold was placed right here. It was allowed to grow, and as it grew, it emitted this penicillin that killed off the bacteria around it, creating this zone of inhibition. And you'll get to learn about that in microbiology class. But for now, we'll just leave it here. You might also be familiar with fungus in the form of yeast and how they help us in fermentation reactions. So in fermentation, we use brewer's yeast to produce our beer, and we also use baker's yeast to bake with in our kitchens. Both of these yeasts are different strains of Saccharomyces cerevisiae, which is, of course, a yeast. Saccharomyces cerevisiae, and cerveza, if you're familiar with Spanish terms, cerveza means beer. So that's how you can easily remember that Saccharomyces cerevisiae is brewer's and baker's yeast. We actually have the downside of fungi as well. Most plant pathogens are actually fungi. This includes our rusts, like the cedar apple rust that we have in our Maryville College orchard right back here in Maryville College Woods. And we also have things that are fungi called blights. If you remember way back when we were talking about allele frequency changes and the potato blight that caused the famine in Ireland, that is a fungus. We also have about 50 to 100 recognized species of fungi that are human pathogens. They cause disease, morbidity, and mortality in humans. One that you might be most familiar with is called Candida albicans. This is responsible for the thrush in people's mouths when you have that white foamy thrush mouth that is caused by an infection by Candida albicans, but also Candida albicans is responsible for yeast infections, both in men and in women. So that's their importance overall, but they also have very important ecological roles. They break down organic matter for nutrient cycling. This is an extremely important role in the environment that is played distinctively by our fungi. They're very good at excreting those extracellular enzymes, and so that is why they are such strong members of the ecological community. They also form these relationships, these intimate associations with plant roots. We have these mutualisms between plants and fungi in the form of what we call mycorrhizae, aka fungus roots. And there's a thing you can do if you're ever gardening or doing some landscaping. Pick up a root and notice how much stuff globs onto it. Okay, so when you're uprooting a plant, you're not just getting the plant roots, you're also extracting with it some fungi that have formed this intimate association with the root called the mycorrhizae. And that mycorrhizae helps influx nutrients because it's, again, excreting those extracellular enzymes, making nutrients readily available not only for the fungus, but also the plant root. So there is a strong benefit to have an association, if you're a plant, with a fungus. Another type of mutualism that you may not be aware of are lichens. Okay, lichens are a mutualism between a fungus, which allows structure, and an algae or a cyanobacterium, which provides food. So when you're looking at a lichen, it's not just one organism that you're looking at what is represented in seemingly one organism, a mutualistic relationship between two different organisms. Our fungus and a cyanobacterium, or just another photosynthesizer. And here are the 3 types of lichens that you'll probably see in your backyard or on campus. We have the crustose lichens. These form flat, crusty patches. You'll most notably see these on rocks. They could be green or white or even red in color, depending upon where you are. We also have the foliose lichens. They're pretty leafy in appearance, and they look like this with these lobed structures like this. You'll see this on tree bark, maybe some rocks. And finally, we have the fruticose lichens. These are even more finely branched, and sometimes they even look fuzzy or furry, like beard or hair. And this is an example of fruticose lichen, okay? So remember, whenever you see a lichen, you're seeing an intimate mutualism between an algae or cyanobacterium and a fungus. It just blows my mind. It's really cool stuff. Okay, so now talking again, focusing on the structure called the hyphae. The hyphae are part of the non-reproductive part of the fungus. It is called a vegetative structure because it is non-reproductive. These hyphae have a tubular shape. So singular form of the word is hypha. Hyphae, A-E, is the plural form. So all of these hyphae have a tubular shape, okay? And it's made of one continuous cell. It's filled with cytoplasm as well as nuclei, okay? Therefore, if this is all one cell, that means that it is multinucleate, meaning there's more than one nucleus inside a single cell. Okay, so sometimes, like for example, over here in panel A, it looks like there are chains of cells. And that is, you know, superficially with your eyeballs, okay, sure, it looks like that. But actually, we have these cross walls called septa. So they're not cell membranes. They are cross walls that separate different compartments of a single hypha, okay? So still it is one cell, but we have septa separating each compartment, or we can be without the septa and be conocytic, okay, without those cross walls. So those are the 2 morphotypes of hyphae that we have represented in our fungus. But either way, whether you are aseptate or septate, you still have a hard cell wall made of chitin. Talking more about different structures, this photo might freak you out. It is a little hard to look at. But if we were looking at the bread mold that forms if you neglect your sliced bread for too long on the counter, this is what forms, okay? So we have this stolon, which is a horizontal hyphae, which is the horizontal hyphae that connect groups of hyphae to each other. Okay, so if this is the surface, it could be a soil surface or it could be the surface of your bread. The hyphae that protrudes up and is exposed and connects 2 different groups of hyphae, that is called the stolon. And then here, notice we have these root-like structures. These root-like parts of the hyphae help to anchor the fungus, and we call these rhizoids. Okay, so not a true root system. Again, these are not plants. They have rhizoids. So it appears to be like the rhizosphere of a plant, but it is not. It is a rhizoid. So we can have bundles and bundles and loads of these hyphae coming together to form an extensive network. This extensive network is called the mycelium, or plural mycelia. But either way, we have an extensive, vast feeding web of hyphae that can protrude in all directions that helps the hyphae grow and expand into more and more physical space. And these hyphae not only go underground, but they're also found inside the thallus, and the cap of those club mushrooms that we see growing outside. So whenever you see this fuzzy white stuff that's forming a web, you might, again, be digging in the dirt in your backyard. You can recognize that easily as mycelia. and hyphae will grow from their tips, which is what we're going to talk about in the next slide, being important for feeding. So these hyphae and the extensive network that they form, the mycelia, facilitates absorptive heterotrophy. So again, fungi get their carbon from organic sources. They get it from dead organic material. So here's an example of a hyphae. It grows from the tip here. The nucleus is going to direct the digestive process. So it doesn't move necessarily, it just tells the tip what to do. So the tip of this hypha excretes enzymes. These are called extracellular enzymes because they are being removed from the inside to the outside of the cell to do its work outside of the cell. Okay. Once these extracellular enzymes find that organic detritus, that dead material, it begins to break it down. Okay. So we have this enzymatic breakdown of substrate, that substrate being the dead organic material. Those products then diffuse back into the hypha. Okay. So from the nucleus, we get the direction to secrete enzymes. Those enzymes act to break stuff down in the surrounding environment, and then the hypha absorbs it back inside. Now, what's extremely interesting is that this vast, extensive network of mycelia allow fungi to be some of the largest organisms on the face of our planet. So it is said that one representative of the Armorillia genus, which is a fungus found in Miller National Forest in Oregon, is the largest organism on Earth. It is 1,000 hectares big and as many as almost 9,000 years old. It is called the humongous fungus, and there's an article for you to read posted on the tartan for you about what this means ecologically and how it just became such a vast, encompassing organism. We also have fairy rings. And I don't know if you've seen these before. I think they're pretty magical and captivating, but it is just an example of what absorptive heterotrophy does to the environment. So we're going to watch this little clip from SciShow that discusses how these form.
A few weeks ago, I was walking through the park and I came across something kind of weird, something that I could not immediately explain. And when that happens, it's usually pretty annoying for me. It was an arc in the grass of greener grass, about 10 feet long, as if someone had very carefully spread fertilizer along a curving stripe of the park. And once I saw it, I started to see more, dozens of them spread across the park. One of them was even a full circle. When I find something just hanging out in the park that I've never heard of and I can't explain, that's frustrating, but thankfully I have Google, and SciShow has a lovely, helpful audience because now I have the answer: fairy rings. Maybe you've seen one of these yourself, an arc of greener grass or an arc of deader grass, or even weirder, a perfect circle of mushrooms. Before we had science, people thought that these might be portals between worlds. If some weird nuisance was occurring in the village, it was likely caused by elves or fairies coming into our world through fairy rings. But alas, this is not the case. And I've already given you a hint as to what they actually are. The key players here are not some elven mischief makers, but fungi. The most common fairy ring mushroom is the Scotch bonnet, and like most fungi, the bulk of its body is its mycelium, and it's underground. The mycelium is kind of like a subterranean tree with thousands of fine branch-like structures that grow outward in a circle. As it expands underground, the mycelium releases chemicals that break down organic matter in the soil nearby, so it has food by the time it grows there. And since by doing that, it's basically fertilizing the soil as it goes, the grass above the leading edge of the mycelium for a short time has more nutrients available than usual, which creates rings of rich, thick green grass. When the mycelium reaches the enriched soil, though, the party is over because the fungus then absorbs all of the nutrients out of the soil and the grass above it dies. And it's at this point, as the fungus matures, that you see the stuff that you probably think of when you think of fungus. Mushrooms begin to sprout, which are actually known as the fungus's fruiting bodies. They're like the fruits of the tree. They generate and release spores that the fungus uses to reproduce. In some cases, unless there's some physical barrier to stop it, a fairy ring can grow for years and cover huge areas. One fairy ring in France was measured to be some 600 meters wide. It's important to note now that fairy ring can also be used to describe a different phenomenon. Redwood groves can also grow in something called a fairy ring, but they're completely different, caused when shoots sprout up from the base of a fallen tree. In the deserts of Namibia, meanwhile, grasses have been found to grow in rings caused by sand termites that live underground. The termites spread out in a circle, eating the roots of all the grasses they come across. This helps the soil retain water, which nourishes the grass at the perimeter of the circle. Sometimes it can feel like science is poo-pooing the mystical and the fantastical, but in my opinion, there's more depth of beauty in the fantasy of the real. That giant fungi reach in circles underground with 600-meter diameters, converting detritus into fertilizer, forming themselves, altering their environment, and interacting on a global scale to form part of this beautiful and complex web of life. But you are entitled to your own opinion as well. Thanks for watching this episode of SciShow. If you have any questions or comments or suggestions, you can find us on Facebook and Twitter, and of course down in the comments below. And if you want to continue getting smarter with us here at SciShow, you can go to youtube.com/SciShow and subscribe.
There's more magic in the real. I love that so much. Okay, so we also have a TED Ed video with the same guy talking about the origin of fungi and how they interact with their environment. And so, as we've already mentioned, the mycelia allow the fungi to cover a large surface area that helps contribute to its type of metabolism that is absorptive saprotrophy. So here is just a depiction of how that fairy ring would form. We have a spore that lands in the center of a lawn, for example. It begins to germinate, and as it germinates, it forms a larger and larger circumference with its mycelia, okay, forming this vast, extensive feeding network. So in addition to this, there's another type of hyphal structure that is extremely interesting, and it is called the hastoria. This is a parasitic type of hyphae. It can trap, like physically trap prey, like plants and animals. And so here is, I think, an electron micrograph showing a small nematode worm that's being physically attacked by this type of parasitic hypha from a yeast. So they're more active than you would think. They're not just, loafing around. They can actually sense their environment. Okay, now moving on to reproductive structures. So we just finished talking about vegetative structures, the hyphae, the mycelium, the hastoria. Now we're talking about reproductive structures. This has to do with asexual and sexual spores. So as I said at the beginning of this lecture, most fungi reproduce sexually and asexually by their spores. But asexual reproduction is the most common type of reproduction. And as with all asexual reproduction, there's no genetic novelty or diversity that's introduced. Therefore, all of the offspring are going to be genetically identical to the parent. What causes a sexual reproduction event is when conditions are poor and nutrients are scarce. So fungi, they want to be producing asexually in a happy, nutrient-rich environment, but when conditions are scarce and things are in the environment pretty downtrodden for them, they produce sexually. And each spore contains a reproductive cell that forms a new organism. That spore is non-motile. Instead, it has to be dispersed by wind. So it's not like a swimming sperm, for example, as we learned about with our bryophytes and our mosses. It has to be dispersed by wind. And this actually helps out because it ensures that species are going to disperse into new geographically novel locations. So this actually helps them spread their genes across a wider geographic area. And so here is what a schematic looks like that represents both asexual and sexual reproduction here. We have that fruiting body with the cap and the thallus. The spores in this type of mushroom are produced along these protrusions called the gills, and we'll get to talk about that at the end of this lecture. But these spores are formed, and once they are ready to go, they can be dispersed via wind. Then they land somewhere else, and once they are about to germinate, we have the hypha that grows underground here, again, growing from the tip. We'll have mating between hypha from different spores. So again, this is a sexual form of reproduction. We have the growth, the continuation here with the hypha continuing to feed through that absorptive mechanism with the emission of extracellular enzymes, and then a young mushroom is going to form, and thereby completing the cycle here. Sexual reproduction is utilized when environmental conditions are poor. Maybe there's not enough space, maybe it's too dry, maybe there's not enough nutrients. What's interesting is there's no male or female fungi. Fungi don't show sexual dimorphism. Instead, they show a dimorphism based upon their environmental conditions as they grow, which is just fascinating. So dimorphic fungi can grow either as a mold form, being a filamentous mycelia, or they can be a yeast form. And the form they take is dictated by the temperature at which you grow them. So here are different representatives of dimorphic fungi. Not all fungi are dimorphic, but here is an example of just a few. We have in the environment, if you were to stumble upon a blastomyces, for example, out in the soil, in the environment, it would look like this. It would have the mycelia, an extensive, vast filamentous network. But if you were to take that blastomyces, grow it in a petri dish at 37 degrees centigrade, it would grow like this. Okay, so it is taking two forms. That is why we call it dimorphic. Di meaning two, morph meaning form. So the process of sexual mating in our fungi happens like this. So we have a haploid hypha, which we represent as 1N, having one copy of the genome. The haploid hypha from two mating types, a positive and a negative, are going to fuse. And this is where we have fertilization, okay? So this forms a hypha with two nuclei. We have this fusion here, and a zygote forms. And the zygote is going to divide to turn into a spore, okay? So this is what sexual reproduction looks like in our fungi when we have the fusion of two mating types, a positive and a negative here. In asexual reproduction, the modes are similar to what we've already learned about in this class. We have fragmentation. This is when part of the mycelium becomes separated from the rest of the main body and begins a life of its own. So we have fragmentation. We also have a phenomenon called budding. This is when a small cell begins to form on a larger cell, and then it gets pinched off. And this is how yeast are able to reproduce. This is budding yeast. So for example, here in this electron micrograph, we're able to see one individual yeast cell. And we have these pock marks or these dimples where previous buds have popped off. And here we have one that's just in the process of popping off or pinching off of the main parent here. So budding is a main way by which yeast are able to asexually reproduce. We also have the production of asexual spores. This is where we have the production of spores by a single mycelium. These spores can grow directly on the hypha itself, such as in penicillium. It could also grow inside a sporangium, and this is a pilobus sporangia with the little spores growing on top here, or finally can grow on those fruiting bodies, so underneath the caps of an amantia, for example. So the fruiting bodies, we've used this term several times, are modified hyphae that make asexual spores. So an upright stalk here If we go back to our bread mold example, the upright stalk is called the sporangiophore, and it supports the spore case called the sporangium. Multiple sporangia, okay? We have different types of fruiting bodies. And again, this will relate directly to the way in which we classify them. So we have fruiting bodies called basidia. Here's an example of a typical mushroom that you would see in the backyard with the thallus and the gills right underneath. And the gills are lined with these structures called basidia that will produce basidia spores. And those are the spores that will be dispersed by wind for future germination in soil. We also have sporangia, as we just talked about on the previous slide, that has the sporangia sitting on top the sporangiophore. And the sporangia, again, produces spores that are able to be dispersed by wind. And then finally, we have the ascus. So we have something that's like a cup-like fungus that has the ascus, and on the ascus structures are the ascus spores, again, able to be dispersed by wind. So the three types are the basidia, the sporangia, and the ascus. Now talking about classification and relationships between each other and us. So as we said at the top of this lecture, fungi are more closely related to animals than they are to plants. And that is made readily apparent when we take a look at our cladogram here with animals on the right and fungi, our closest living relatives on this cladogram. And these fungi are classified by the different reproduction structures and their spores. So we just learned these words on the preceding slides, basidia, sporangia, and ascus. These go on to name the different phyla that we have within the fungi. So we have Basidia mycota represented here. The sister taxa to the Basidiomycota are the Ascomycota. And we also have the Zygosporangia over here. We also have different types like the Glomericota and the Chytromycota. This is a pretty newly recognized phylum, and this one is microscopic, so you may not have heard about it before. But we're going to go through each of these and show you representative types and what they're used for and why they are important. So first we have the Basidia mycota. These are called the club fungi. This includes our mushrooms, our toadstools, our puffballs, our stinkhorns, our rusts, and our smuts. So this is probably the type of fungi with which you are most familiar, are the Basidia mycota. Next we have the Ascomycota. These are called sac fungi. This includes our cup fungi, our morels, our truffles. This also includes our yeast, so our baker's yeast and our brewer's yeast. These can also be plant parasites as well, like our Dutch elm disease or chestnut blight. And these ascomycetes are able to reproduce sexually and asexually. And as we've already mentioned, the main structure for which they are named is the ascus, and this is a sac that makes the ascospores in sexual reproduction. Next, we have the Glomericota. This is newly established, but even with that considered, we have 230 species. So it's pretty diverse for a new phylum, and this is formed in close association with roots of trees. We have the Zygomycota as well. This is called a sporangium fungi, and this is what we know and love as mold. So when you have your moldy strawberries sitting so sad in your fridge because you forgot to eat them this week, you can say, hey, I know what that is. That is Rhizopus, and that is part of the Zygomycota phylum. The more you know. The Zygomycota also includes some blights, and they have hyphae without any crosswalls, so they are aseptate. They're able to grow rapidly, and of course, we associate rapid growth with our molds, specifically Rhizopus, for example. Finally, we have the Chytromycota, or the chytrids. These produce motile spores that are able to move around. These are mostly saprobes and also parasites. Okay, so here is an example of a chytrid fungus that's attacking a small Daphnia water flea, which is an aquatic invertebrate. We can see it being attacked underneath the microscope. But more visibly with our eyes, we're able to see the devastation and destruction that chytrid fungus has wrought on the amphibian communities. Here depicted with this poor little guy that has this fungus just coating his entire body. And what's extremely sad is that chytrid fungus is responsible for the extinction of the Panamanian golden frog in the wild. I think there are some representatives that are still in zoos, but this fungus just ravaged the entire species of the Panamanian golden frog when I was in college. And so I was extremely sad to hear that it's officially been decreed extinct in the wild now.